Record just now. Good morning, everybody. Welcome to Life Sciences Grade 10. My name is Tutuzile Gopega. We are discussing cell organelles, continuing with that topic, but today we'll be dealing with ribosomes and Golgi body. But before I do that, guys, I am so sorry about yesterday. I was having so much fun with the game. I didn't even check the time. And then we got cut off because Zoom cuts us off if we go above the time limit that is set. So please forgive me regarding our game. Next time I'll be very mindful of the time because it was so fun. We didn't even get to see the winners and all of that, and I was really having fun with it. Thank you so much for your participation as well. I will set it up again and then we will play for the rest of the topics because I only covered about 2 lessons for that one. I'll also do it as we do our revision, because we're also going to be starting with assessments. I'll incorporate it when we do our revision. I think it will be a helpful way for you guys to revise. But this time with the fewer questions so that we can be able to finish quickly before time is up. I promise, but please forgive me about what happened yesterday. I couldn't even log back in to tell you guys what had happened. So we just got cut off just like that. So yeah, we've learned our lesson, all of us. Next time I will do better. Then we've got topic today. At the end of this lesson, you should be able to know ribosomes and Golgi body. Ribosomes. What are ribosomes? Remember, we've been discussing ribosomes when we were discussing all the other organelles. We also have ribosomes. Ribosomes are composed of RNA and protein. They are composed of RNA and protein. They occur in the cytoplasm and are the sites where protein synthesis occurs. So that's the thing about ribosomes. They are composed of RNA and protein. Remember that what we've been learning about so far is the DNA. Now we see RNA. They occur in the cytoplasm. These are the sites where protein synthesis occurs. So when the various stages of what's this gene information control, what happens is you'd first have replication, then you'd have transcription. Then you would have what we call translation, where it's from mRNA to what? To the proteins. Now you are making proteins. That is the process of translation where protein synthesis occurs, where now from these amino acids, you are making proteins. That process occurs whereby the genetic information will leave the nucleus and go to the ribosome, where protein synthesis would take place. You'll see when we do the part about DNA and RNA and those genes and protein synthesis, the whole topic of protein synthesis, you'll notice from the diagrams how the DNA, after the 2 steps of replication and transcription, it will move out of the nucleus through the nuclear pores to the ribosomes where protein synthesis will occur. And then ribosomes may occur singly in the cytoplasm or in groups, or maybe attached to the endoplasmic reticulum, thus forming the rough endoplasmic reticulum. That's where we've seen the ER. Remember when we saw with the last topic we dealt with was when we were discussing the endoplasmic reticulum. And we've seen that the rough ER has lots and lots of ribosomes around it, giving it that rough structure. So that shows you how they can occur. They can occur singly in the cytoplasm, so they could just be on their own there. Or you'd find them around the rough endoplasmic reticulum. Ribosomes are important for protein production, mainly because that's where they are the site of protein synthesis. It occurs there. And then if without it, then we wouldn't be able to make protein in that cell so efficiently. Together with a structure known as messenger RNA, this is a type of nucleic acid. We will discuss more about the different types of these nucleic acids when we discuss the topic of genes. The messenger RNA called mRNA, the ribosomes would form a structure known as a polyribosome, which is important in protein synthesis. So know this for now. It will be very useful and will make more sense when we deal with genes. So these here are the polyribosomes. These are the polyribosomes. You've got what? The plasma membrane. Remember the membrane? And then inside you have the free ribosomes. These are the ones that are just free on their own. They occur singly. Remember, you can find them in the cytoplasm just occurring on their own. So you can find them like this, or you can find them around the endoplasmic reticulum, which I showed you the day we discussed the rough ER. And then this here, the polyribosomes, it's when you have the ribosomes with what? With the mRNA. So when they are together, they form the structure called polyribosomes. So remember these structures. I'm showing you individually so that you'll be able to recognize them when you see the whole diagram. So this is a bigger picture of the polyribosome over here. The mRNA with the ribosomes around it. This is a diagram of several ribosomes joined together on a strand of mRNA to form a polyribosome. So you see ribosomes like attaching and binding together with various structures. Okay? So I think that will help with the functioning synergistically. So then now we are moving on to the organelle called Golgi body. This Golgi body, this organelle here, is found near the nucleus and endoplasmic reticulum. The Golgi body consists of a stack of flat membrane-bound sacs called cisternae. So it has it consists of a stack. There are flat membrane-bound sacs, lots of them. The stack of them that are called what? Cisternae. The cisternae within the Golgi body consists of enzymes which modify the packaged products of the Golgi body. These are the proteins. So it consists of lots of enzymes. They modify these proteins. This is an image or diagram, the schematic diagram of the Golgi body. So I think now if you can just go through all the lessons, you'll be able to identify. Let me tell you now, when assessments start, I will be giving you these structures and I will ask you to label them. Or I'll just give you a complete cell and I'd ask you to label each and every part of that cell. And that question would go for about 10 marks or so. So please be mindful of these structures and understand their functions. It is important that you are able to identify these structures and know their functions. This is a diagram that's showing Golgi bodies found in animal cells. There's your Golgi body. You see those stacks? They are the stacks here, the cisternae. These are the stacks. Stacks because they are stacked together. That's why we say it like that. So they are Golgi bodies. This is the micrograph. Remember micrograph? Taken using the transmission electron microscope of a Golgi body. Visible as a stack of semicircular black rings near the bottom. You see here, this is the Golgi body when you look at it under a microscope. These are the stacks, the cisternae, lots and lots of them. So you can clearly see that this diagram here, the schematic diagram, is based on such images. So it's pretty much accurate, as you can see. Ne? Did you know the Golgi body was discovered by the Italian physician Camillo Golgi? It was one of the first organelles to be discovered and described in detail because it's large in size, made it easier to observe. So because of the size, you see, if you I'll show you a complete cell structure, then you're able to compare accurately and see. But because of its very, very large structure, then it's easy to view it under a microscope. That is why they were able to view it and be able to identify and describe it in detail. It was discovered by the Italian physician Camillo Golgi. This gives it away. You see the name, they name these things after themselves. Camillo Golgi. So when I ask you who discovered the Golgi body, if you have forgotten this, you can even say C. Golgi, for goodness sake, but you can't forget the Golgi part because the answer would be in the question. So remember this when we get to that part. And then functions of the Golgi body. What are the functions of the Golgi body? It is important for proteins to be transported from where they are synthesized to where they are required in the cell. So they are synthesized where? They are synthesized in the, remember protein synthesis, the site of protein synthesis. I said it in this lesson. What is the site of protein synthesis? This is a question posed to everyone in class. So if you could please give me an answer, everybody. The site of protein synthesis. That is my question to you. What is the site of protein synthesis? I discussed it in today's class. If you can't answer me, it means you're not listening to me. Whoever gets this right, I'm getting them a prize. That's what I'll do actually when we do the Kahoots games. Whoever wins, I'll give you a prize. Ne? We can discuss together what the prize should be. But I'm committing to giving you prizes. Now, first up, tell me the site of protein synthesis. This one who gets this right will get data. Or even in, I don't know, data or what? I don't know what else we'll discuss. What is the site of protein synthesis? Guys, should I call you out by your names? What is the site of protein synthesis? I discussed it with you today. I'm giving you like a few seconds to answer. Yo, I'm shocked. No one knows the answer. Guys, let's go back here. Today's lesson over here. Ribosomes are composed of RNA and protein. They occur in the cytoplasm and are the sites where protein synthesis occurs. So protein production, protein synthesis occurs in the ribosomes. Ribosomes. And then now we are discussing Golgi body and the function of the Golgi body. And it's mentioned here that it is important for proteins to be transported from where they are synthesized to where they are required. Same way when you bake at a bakery, you don't want to bake to keep the goodies there. You want to sell them and transport them to people who actually want those goods and want to buy them, right? So protein synthesis is synthesized, protein is synthesized at one point, which we have just learned, it is the ribosomes. And then after the synthesis of those proteins, they are then transported to various parts of the body of the cell where they are required. And the Golgi body is responsible for that. It is responsible for transporting of the protein that was synthesized in the ribosomes. Like we said here, the organelle responsible for this is the Golgi body. The Golgi body is the sorting organelle of the cell. So it sorts, it ensures that this needs to go to this place, this needs to go to this place, and so on and so on. Proteins are transported from the rough endoplasmic reticulum to the Golgi. Why the rough endoplasmic reticulum? because that is the one site you will find lots and lots of ribosomes there. Remember, ribosomes are all around the endoplasmic reticulum, making it this rough structure. So the proteins would move from where they were synthesized in the ribosomes there on the rough endoplasmic reticulum. And then they would be transported by the Golgi board, from there to the Golgi bodies. So they would move from the rough ER to the Golgi body. And then in the Golgi bodies, when they get there, these proteins will then be modified and packaged into vesicles. So they'd be modified, you'd sort them. This protein is needed, protein A is needed in this part. So it's packaged to move along with those parts that are going to that site. This protein is going to this part, it will package like that. Same way, if you place an order, let's say you place an order through Take-A-Lot, various other people are placing their orders there. So you place the order, and then what would happen? The people at the warehouse would sort out the things. Things that are going to Johannesburg South will be packed probably in one vehicle because they are going to one place anyway. So this vehicle will drop them off. Those that are going to Bloemfontein will be packaged on that side, and then they will be dropped off at that area. Those going to East Rand will be packaged, and then they'll be dropped off in East Rand. Those going to Mthatha, things like that, they would be packaged. So that's what happened. The Golgi body is like the warehouse here. It is where all the sorting takes place, where you check the addresses, what is needed, which address. Okay, I package it for them. Then you put them into the vesicle, which will then be the one transporting. You understand, right? That's the whole process. That's how it functions. Think of it like that, then you understand it better. The Golgi body therefore receives proteins made in one location in the cell and transfers these to another location within the cell where they are required. So they what? They are made in one location in the cell and they are transferred to another location within the cell where they are required. For this reason, the Golgi body can be considered to be the post office of the cell. So yeah, I should have made the post office example instead of the warehouse. Post office, there are various letters coming through to them. What do? They will sort, still according to, it's really the same examples really. They sort them according to the different addresses. Those going to Joburg South, go Joburg East, Joburg West, they'd be in different vehicles that would transport them to those areas. But before they can be transported, what needs to be done? Some sorting needs to be done so that you can be able to group them together according to the locations they are supposed to be going to. So that is the point of Golgi bodies. Think of post office, think of the warehouse, things like that. That's the general description of this. So guys, I hope it's clearer now. I hope you are paying attention. Please, please. This is for you at the end of the day. You need to pass. Any questions? Feel free to ask me any question you may have regarding life sciences. Did you also, okay, let me tell you, did you receive the newsletters that next week assessments are starting next week? Receive the newsletter. If not, please sign up for the newsletter, guys. Remember, there is the site where you can sign up for the newsletter. Go to www.africateamgeeks.co.za. Over here, you can be able to sign up for the newsletter. And you would need to be able to sign up as well so that we can even get information about assessments. The timetable is already posted there about when assessments will be happening. So check this out. And then that other one is my personal email address for your queries. If you have any questions you want to ask me about life sciences over here, you are most welcome to ask me and I will engage you. So feel free to ask me. Send me an email. If you're too shy to ask in class, send me an email. If you're doing certain work, maybe in your school they send you homework online and you are confused, ask me again. I will help you. As long as it's life sciences related, I will gladly assist. So there's my email address. But remember to go sign up for the site here. Sign up so that you can get access to more information. You can always be on the loop about things that are happening. Anyway, any questions regarding today's lesson? Any questions regarding yesterday's Kahoots? Let's talk about Kahoots. Did you understand it? Those who didn't participate, were you able to see how it goes? And are you at least interested in joining next time? Can you talk to me? Can we unmute you? Itumeleng, please unmute all of them. If they will talk to me, I don't know. Please talk to me now.
They can unmute themselves.
Okay, guys, you can unmute yourselves. Talk to me.
Ma'am, when can you do the Kahoot again?
I don't know. We can do it sometime this week. What day is it today? It's Wednesday, right? I think
Yes,
we'll do
ma'am.
it Friday. Yeah, we can set it up for Friday. But people must participate. At least the ones who participated yesterday. Did you enjoy it? Was it okay? You learned, ne?
Very nice.
Oh, okay. I'm glad. I'm sorry it got cut off. I'll make sure next time I'm mindful of the time.
Okay.
And then everyone else, BMAX, are you okay? Junior, Lois, Xolo. Okay, you will join on Friday. All right. Okay, then I guess I'll see you tomorrow if you don't have any questions.